Welcome to our seminar today. It's my pleasure to introduce our speaker today, Professor Yang Dan from University of California, Berkeley, both because she's a fantastic neuroscientist and a leader in the field, but also because I did my postdoctoral work with her 20 years ago. Sorry to date you and me.
23 or no, yeah. 20, yeah, whatever.
Like in fact, she helped me make my transition from engineering into neuroscience. So for that, I'm always grateful. So Yang did got her bachelor's in physics from PKU, from Beijing University. She went on and made a transition in physics, sorry. Her bachelor's in physics. She went on to do her PhD in biology and neuroscience at Columbia University, where she studied synaptic plasticity and signaling mechanisms. She then made a transition into the visual pathway and systems neuroscience in her postdoctoral work at Rockefeller, where she did work there and then transitioned to Harvard University, working with Clay Reed, who we had him as a speaker a year ago or so, where she did fantastic work in the visual pathway. She then went on to join the faculty at Berkeley in molecular and cell biology in the late 90s, and that's where I met Yang. And I remember at that time, when she got hired, we saw many different seminars coming through, and there were all these kind of boring neuroscience talks that we've all sat through. And she came and gave this just wonderful talk. It was really exciting, and for me, it was an exposure to a side of neuroscience I didn't know about. It was very, very exciting. And she went on to start her laboratory there in Berkeley, and working on a wide array of different problems. And it was really fun to watch her build a laboratory and be part of that startup culture at that time, to see how different projects get formed and so on. So she had a real knack for sniffing out really exciting problems. If you've looked at her background, at her papers, you see that she's spanned a lot of different areas, from synaptic plasticity to visual pathway, both coding problems as well as mechanistic things, on to brain state and other really interesting things that the field has gone on to follow, and then more recently in sleep. She is, I have this just in case I forgot, the Paul Licht Distinguished Professor, as well as Howard Hughes Medical Institute investigator, and is a member of the National Academy of Sciences. So it's really a pleasure to have you here, very exciting, and I hope you all welcome her, and I'll turn it over to you. Thank you so much for coming.
Thank you. So I'm now wearing this microphone. Can you guys at the back hear me at this volume? No? Did someone say no? Yeah? Okay. So I'll try to scream, and then if you can't hear me, just wave your hand. And then the other thing is that I know that you guys, a lot of you are engineers, so if I'm not explaining things clearly, you can feel free to interrupt me. I don't have to finish the talk because it doesn't matter, right? So first, thank you, Gary, for the nice introduction. Gary was actually my very first postdoc. So without Gary joining my lab, my tenure would have been very questionable. So, it's very important. Okay, so I'd like to tell you about our work on sleep. So this is something that my lab started about 9 years ago. So we're still kind of new to this field. Sleep is an essential innate behavior, right? It's innate because we know how to do this when we're born, right? We don't need to learn how to do it. It's essential because, as far as we can tell, all animals sleep, including flies and worms, and even animals without a central nervous system, including the jellyfish, with only sort of a spread out neural net. So for non-mammalian animal models, sleep is usually detected by a lack of movement. So they have to stay still for a period of time. There's usually a threshold duration. That's how you score a sleep episode. And also, when you use a mild sensory stimulation, you fail to evoke a motor response. But everything is measured motorly. And for mammalian animal models, we measure EEG and EMG. So when we fall asleep, there's a very clear reduction of skeletal muscle tone measured by EMG. And then based on EEG, we can distinguish rapid eye movement or REM sleep, and also non-REM sleep, which is also called the slow wave sleep, because you see these large amplitude slow waves during non-REM sleep. So for humans, we measure EEG, EMG, and also a few other things like heartbeat and also breathing. So when we fall asleep, right, there's a dramatic reduction of somatic motor activity measured by EMG, and also there's a reduction of the autonomic motor output, like your heart rate goes down, breathing rate goes down. So the point is that falling asleep is not just about changes in the brain state, as you can measure in the EEG, but also it's about the reduction of the different types of motor output. And so what I hope to convince you today that when you look at the neural circuit controlling sleep, actually a lot of them are also closely associated with these motor circuits. And that's why I have this sort of strange title for my talk. Okay, so pioneered by Moruzzi and Magan, who discovered the ascending arousal system, we actually know a lot about the neural circuits controlling wakefulness and arousal. And so now we know that there are a lot of these well-known neuromodulatory systems, such as the cholinergic system and also monoaminergic systems, such as noradrenaline, serotonin, histamine, and dopamine. There are also some neuropeptide systems. The best known one is the orexin/hypocretin system, because if you lose that, you have narcolepsy. In contrast, we know much less about the neural circuits controlling sleep. So according to the textbook, there's a single center in the anterior hypothalamus. It's called the preoptic area, or POA, where a subset of the neurons promote sleep by inhibiting a lot of these well-known wake-promoting neural systems. So indeed, using retrograde labeling from some of these wake-promoting centers, which we know a lot about, we were able to identify a subset of the neurons in the POA that actually promote sleep. But it turns out this is far from the whole story, because about 4 or 5 years ago, we and others have identified some sleep neurons outside of the POA, suggesting that the network is actually much more distributed than this model suggested by this textbook model with a single sleep center. So to get an idea of what this distributed network might look like, we decided to do a whole brain screening for sleep neurons. So we have two basic criteria for, actually maybe I should use the cursor because some people are looking at that screen. So we have two basic criteria for sleep neurons. First, they need to be sleep promoting, right? So the idea is that their activation should increase sleep and their inactivation should decrease sleep. Secondly, they should be sleep active, meaning that they need to be active at the right time to do their job. And for screening for sleep neurons, we also have two corresponding strategies. We could either screen for sleep promoting neurons and then see if they're also sleep active, or we can screen for sleep active neurons and then see if they're also sleep promoting. So usually the sleep neurons, when we call them the sleep neurons, they need to satisfy both criteria. So I'm going to give you 1 or 2 examples of each strategy that we have used. So to screen for sleep active neurons, we use this technique called the false trap, developed by Li Chun Luo's lab at Stanford University. So we cross 2 mouse lines together. One of them expresses CRE-ER under the FOS promoter. So FOS is an immediate early gene that's activity dependent. Usually when a neuron has been active recently, they turn on the FOS gene. And in this mouse, they should also turn on CRE-ER. And then the other mouse expresses GFP in a CRE-dependent manner. So we divide these two, this cross mouse line into 2 groups. One of them, we call them the SD, the sleep-deprived group. So what we do is to inject tamoxifen during sleep deprivation. And the idea is that wake-active neuron, right, they should be active during sleep deprivation because we keep the mouse awake during this period. So they should express CRE-ER. And then the injection of tamoxifen should cause CRE-ER, which is a molecule, to enter the nucleus. And then this CRE is a DNA recombinase, right? So when it enters the nucleus, it's going to do something to this other gene, right? And so it's going to turn on GFP in a permanent manner. So we can label the wake-active neurons because we turn on the GFP gene. And then in this other group, we call it the RS group, we're first going to sleep deprive them for 6 hours to make the mouse really sleepy. And then we leave them alone. We basically stop harassing them so they enter recovery sleep, right? And then when we inject tamoxifen, that's going to turn on the GAP in these sleep active neurons that are active during the recovery sleep. So this was done by mostly Zhe Zhang with the help from Peng Zhong. So Zhe has left my lab to start her own lab in Shanghai. So when we compare these two groups of mice, we saw that in some brain areas, right? So these are different brain areas. So in some brain areas, we saw more GAP labeling in the RS group. So those are shown here. And in these other brain areas, we saw more GAP labeling in the SD group, which is hardly surprising. But when we looked at this top candidate, right? This is the region with the highest relative expression of GAP in the RS group, right? So these are the putative sleep neurons. That region is shown here. It's basically the ventrolateral part of the periaqueductal gray. We call it the PAG. So as you can see, there are more GAP labeling in the RS group than the SD group. Now, on the other hand, even though there are a lot more neurons expressing GAP, but this is still a very small minority of all the cells expressing in the ventrolateral PAG. So we have to figure out, who these neurons are. And in order to do that, we did gene profiling. So the reporter mouse line that we used expresses not only GAP, but also this molecule called L10A, which is a ribosomal protein that's normally attached to mRNA because its normal job is for RNA translation. So if we use an antibody to pull down GAP, we can also pull down L10A together with the mRNA attached to it, so we can do sequencing. So that's what the data look like. This is called the volcano plot. The x-axis is the relative expression level of the gene, right? So each dot is a single gene, right? So this is the relative expression level of that gene in the putative sleep neurons, the GAP expressing neurons, relative to the entire chunk of tissue that we cut out. Now the vertical axis is the p-value. So when we zoom into this red box here, right? So this is these are the genes that are highly significantly enriched in the putative sleep neurons. You see a whole bunch of genes. So we decided to focus on neuropeptides for two reasons. One of them is that we know that neuropeptides are important for signaling. Secondly, there are just a lot of Cree mouse lines associated with neuropeptides. So in this box, we saw 3 neuropeptides, and among them, we found that this gene called CALCA, which is calcitonin gene-related peptide alpha, right? The name doesn't really matter. But anyway, so this CALCA gene, the expression shows a high overlap with the GAP labeling of the putative sleep neurons. So we decided to focus on this CALCA marker. So first, we wanted to make sure that these CALCA neurons are indeed sleep active, even though they are labeled by C-false, right? But false is a convenient but imperfect label for activity because it also depends on other factors. So we wanted to make sure that these are really sleep active, and for that, the only way to do it is to actually record the activity. So for that, we did something called the optrode recording. So optrode consists of an optic fiber, right? You see this little optic fiber here, surrounded by several bundles of tetrodes or stereotrodes or whatever. So we tag the CALCA neurons with channelrhodopsin by injecting the Cree-dependent channel expressing virus into the CALCA Cree line generated by Richard Palmiter's lab. So here you can see that we got this nice unit, right? So you see this clean single unit. And every time we turn on the laser briefly, right, 5 milliseconds, the little blue dot here, you see a spiking, right, with a very short latency with a high reliability. So we're pretty sure that this is a unit that expresses channelrhodopsin. Therefore, it should be a CALCA expressing neuron in this mouse line. So once we identify a unit like this, we turn out the laser and just do recording of the spontaneous activity. So here, this is the EEG power spectrogram. So if you see a lot of broadband, low-frequency activity, this is probably a language that speaks to you, the engineers, right? So if you see broadband activity at low frequency, chances are the animal is in slow wave sleep, right? A lot of high amplitude slow waves. But if you see sort of a theta band at about 8 hertz, the animal is either awake or in REM sleep. And you distinguish those based on EMG, right, which is the second trace. Oops, I lost my tracer. Yeah, the second trace here. And then this is the firing rate of an identified calcaneuron. So you can see that this neuron fires at a high rate whenever the animal is in non-REM sleep, right, this orange period. And the firing rate is low during either REM, right, the blue period, or wake, the gray period. So here is a summary of more than 30 identified calcaneurons. Each line is one neuron, right, across the 3 brain states. And we also show our data in this 2-dimensional plot. So the horizontal axis is the firing rate difference between non-REM sleep and wake, right, normalized by their sum. And then the vertical axis is the firing rate difference between REM sleep and wake. So the identified calcaneurons, the blue dots, are mostly on the right-hand side, right, indicating that these neurons fire more during non-REM sleep than wake. But they don't seem to have a consistent firing rate difference between REM sleep and wake. Now, if you look at the gray units, so these are recorded with the same optrode, right, in the same area, but these are not driven by the laser. So we don't know who these neurons are, but at least they're in the same area. But you see that many of them are on the left. So these are wake active neurons. So based on this, we know that the majority of the calcaneurons are non-REM active, right? So this is our first criterion, right? So it basically confirms the CFOS experiment. So now the question is, are they non-REM promoting? And so for that, we did optogenetic activation. So here is sort of a typical experiment. We turn our laser on for 2 minutes per trial, right? The blue shading period. Semi-randomly every 15 minutes or so. And so, at the same time, we were recording the EEG, EMG, and the color bar is just the computer automatic score of the sleep stage. So this is a summary of all the trials from all the mice that we tested. It's basically the laser-triggered average, right? So as you see, when we turn on the laser at time 0 here, so the laser is for 2 minutes, the blue shading here, we see a very immediate and clear increase in the probability of non-REM sleep, right? It goes from the baseline about 60% to about 80%. At the same time, there's a reduction of both wakefulness and REM probability. And when we inactivated these neurons using IC++, which is basically a mutated channel Dobson, that's a chloride channel, we saw a decrease in non-REM sleep and increase in wakefulness. So in addition to this kind of acute optogenetic activation or inactivation, which is just 2 minutes per trial, we also did a DREAD experiment, which is slower, longer term. And you can see that the GQ-mediated activation caused an increase in non-REM sleep for about 2 hours. And the GI-mediated inactivation caused a decrease in non-REM sleep. So based on both optogenetic and chemogenetic activation and inactivation, we know that these calcaneurons are non-REM promoting, which is our second criterion. So here we're just using this neuropeptide calca as a marker, right? But it turns out that in these subcortical regions, most of the neurons, they do different things, right? They use these neuropeptides, but they also have, these traditional neurotransmitters. So it turns out that the calcaneurons, about 60% of them are also glutamatergic, and the other 40% are cholinergic. So without showing you the data, I'll just tell you that we think that the sleep promoting effect is mostly mediated by these glutamatergic subpopulation. The cholinergic neurons, they are actually the preganglionic parasympathetic neurons, and their main their main job is to constrict the pupil when you activate the cholinergic neurons. So they're kind of related to sleep because when we fall asleep, the pupil size also constrict, but their main job is not to promote sleep per se. Okay, so these are glutamatergic neurons that promote sleep. And it turns out that in this region, they're not the only glutamatergic neurons. We also found another population, and these neurons actually express another peptide called CCK. So the CCK neurons are almost 100% glutamatergic, and they're entirely separate from the calca neurons. So in this case, the red neurons are calca neurons. So you can see that they're sort of sitting on the two sides, and the CCK neurons are sitting exactly along the midline, right? So they're entirely separate. And so when we activated the CCK neurons, we also saw increase in non-REM sleep, and their inactivation causes a decrease in non-REM sleep. So the CCK neurons are also non-REM promoting. So we have these two populations, right? So they're sitting right next door to each other. And then the question is, do they talk to each other? And so we did some slice experiments, and it turns out that, so the way that we do this is that in this case, we use CCK Cree mouse, so we can express channel rhodopsin in the CCK neurons, and then we patch, so this is slice recording, and when we patch on a calca neuron, we know that because we suck out the cytoplasm from the neurons that we patch, and then we do RT-PCR to confirm that there are, calca neurons. So in this case, you can see that activating the CCK neurons causes a big excitation, and that's blocked by CNQX, which is a glutamate receptor blocker. So we know that there is glutamatergic synaptic excitation. Conversely, when we activate the calca neurons and record from the CCK neurons, there's also excitatory input. So that just says that these two populations of neurons, they both promote non-REM sleep. They actually talk to each other, right? Which is kind of interesting because this seems to be a way that they can reinforce each other, right? So that they can stay active during non-REM sleep. Okay, so just a very quick summary, right? So what we found so far is that we have found in the VL pack, there are these calca neurons and CCK neurons. They talk to each other. And so they promote sleep. And again, without showing you the data, I'll just tell you that they actually project to the POA, which is the textbook sleep center, right? So the POA sleep neurons are GABAergic. These neurons promote sleep by inhibiting these wake neurons. And then the VL pack neurons, these are excitatory, and they promote sleep. And they do this so this effect seems to be mediated by their projection to the GABAergic neurons, right? And so they seem to be like upstream of the GABAergic sleep-promoting neurons. And then also we found this other region in the medulla, very, very posterior. These are also GABAergic neurons that promote sleep. And the calca neurons also project to the GIV to promote sleep. Okay, so now I'm going to switch to this other approach that we have taken, right, which is to screen for sleep-promoting neurons. So we did this based on a very simple-minded logic, right? So if you don't, the technical details doesn't matter, but hopefully you understand the logic, which is very simple. So we're going to start with the wake-promoting neurons. We actually know quite a bit of them. And what we're going to do is to use retrograde tracing to find their inhibitory input. Because if you're a neuron that inhibit a lot of the wake-promoting neurons, then perhaps your activity can promote sleep, right? Because sleep and wake, these are like incompatible brain states. And once we find these inhibitory sleep-promoting neurons, we're going to trace one more step back and look for their excitatory input. Because if you're a neuron that excite other sleep-promoting neurons, then your activation could promote sleep, right? It's a very simple kind of sign. Either you keep the same sign or you reverse the sign, right? So that's our logic for screening for sleep-promoting neurons. So this was done by a very talented postdoc, Chen-Yan Ma, in the lab. So Chen-Yan targeted a whole bunch of wake-promoting neurons. Like I said at the beginning, we know a lot of them already. And so she was looking for brain regions with relatively broad GABAergic innervation of these wake regions. So this is her first batch of screening. These are the brain regions that she targeted, the wake-promoting populations, including histaminergic, noradrenergic, GABAergic, and glutamatergic, and cholinergic, right? Basically a whole bunch of them in 7 brain regions. And then these are the brain regions where she found a relatively broad GABAergic projection to these wake-promoting populations. So this includes the POA, right, which is the textbook sleep center. But in addition, actually, in this limited screening, the top candidate that Chen-Yan found anatomically is the CEA, which is the central nucleus of amygdala, right, which is normally people think of it as sort of emotional regulation, right? So basically, this is an example of each of the 7 populations that Chen Yan targeted. And in each case, right, her rabies retrograde tracing shows the labeling in this tiny area, right? So you see this little white box here. And if you blow it up, right, you see a lot of the green, which is the rabies virus labeling. And red is basically the marker for GABAergic neurons, right? So in each of these cases, you see a very strong overlap between the rabies labeling and the GABAergic neurons. So again, but the labeled neurons is still a small subset of the GABAergic neurons. So we have to figure out who these neurons are. And again, we did gene profiling, right, using the same technique that I explained earlier. In this case, if we look at the highly enriched genes in this box here, again, we saw 3 neuropeptides. And it turns out that when we compare the expression of these 3 peptides, 2 of them overlap by more than 90%, and 2 of them overlap by 50% or so. And it turns out that this peptide NTS, which is neurotensin, seems to be the best marker because it labels a lot of the rabies labeled neurons. And also NTS is pretty specifically expressed in the CEA, but not in the surrounding brain regions. So we decided to focus on NTS. And again, we used the NTS Cree line. So I'll just go through this very quickly. Activation, right, through channelrhodopsin causes an increase in non-REM sleep, decrease in wake and REM. Inactivation causes the opposite effect. And then, so the CEA NTS neurons are non-REM promoting. We also did opto recording, right? And so here's a summary, right? And you see most of the identified blue NTS neurons, most of them are in the upper right quadrant. So these neurons are actually more active during both REM and non-REM sleep compared to wakefulness, whereas the unidentified gray dots are all over the place. So most of the NTS neurons are also sleep active. Okay, so here we have found a population of inhibitory sleep neurons in the CEA, right? So as promised, we're going to trace one more step back and look for their excitatory input. So Chen Yan mostly found 2 regions with very strong glutamatergic projection. And so one of them is this region here, right? So you see the rabies labeling, and this is VGLU2, which is a glutamatergic marker. You see the overlap. So what is this region? So this is the Allen atlas. And it turns out that this green stripe here, right, with a lot of labeled cells, is basically this sort of vertical stripe right between the border of the thalamus, which is this red region here, and the rest of the brain. And I think that this is the midbrain, right? So it's not the entire thalamus, right? It's like this sort of vertical strip, right? And we don't even know what to call that thalamic region. You look at the atlas, they gave these weird names, and in the literature, there's very little consistent reporting of what that part of the thalamus does, right? So again, we have to find a marker. Now, in this case, we just made a lucky guess, right? Because in the CEA, NTS seems to be such a great marker. And we said, well, could NTS be the marker here? And so when we looked at the Allen Institute in situ hybridization database, it turns out that NTS is exactly expressed in this vertical strip. And when we looked at NTS expression with the rabies labeling, and again, we saw a very high percentage of overlap. So it seems like NTS is also a good marker for the glutamatergic neurons. And indeed, when we activate these glutamatergic NTS neurons, we saw increase in sleep, and inactivation causes decrease. Okay, so now we have 2 populations, right? One glutamatergic and the other GABAergic. And yet the common denominator is neurotensin as a marker, right? And so, I gave this talk. Actually, the paper just came out last year, but I've been giving this talk for more than 2 years, right? Just to show you how long it takes for the publication process, right? So initially, I would just present this, and the question I always get is that, what's the role of NTS, right? And so for a long time, my answer was, I don't really care. All we need is a marker, right? As long as I get the right Cree line, I don't care the rest, right? But if you're asked this question like 20 times, you always give this crappy answer. At some point, you're like, okay, maybe we should do the experiment. So finally, we did it, right? So the way that we did it was to use CRISPR-Cas9 technique, right? Once people figured out this, it turns out the implementation is pretty easy, right? So what Chen Yan did was to design this very complicated construct, but basically it expresses a fluorescent marker and channel rhodopsin, but also the guide RNA for whatever molecule you want to knock down, right? So the control is basically a LAC-Z, it's a random gene, right? And then the other one that's interesting is NTS, right? So if you use the guide RNA against NTS, then Cas9 is basically gonna damage the gene so that you can knock down the expression of NTS. So here you can see that in the control, right, you see the mCherry expression means the virus is working, NTS is expressed. But when you express NTS guide RNA, you can really knock down NTS expression here. And so, so you can do this in the CEA or in the posterior thalamus. Now, when we did this, right, in the control case, which is this white bar, channel rhodopsin activation still causes an increase in sleep, right? So this is just a repetition of the experiment I showed you earlier, except that we also express this control guide RNA. But when you knock down NTS, this black bar here, you see that the REM increase is greatly reduced. Not only that, right, when we express the guide RNA that knocks down GABAergic transmission, we also saw a decrease in the non-REM promoting effect. Now, in the posterior thalamus, you can knock down NTS that basically wipes out the non-REM increase. And if you knock down glutamatergic transmission, you basically also eliminate the non-REM promoting effect. So the conclusion of this is that both the NTS peptide and the traditional transmitter, is either glutamatergic or GABA, are important for the non-REM promoting effect of these neurons. Okay, so here we have NTS that already labels two populations, right? Glutamatergic and GABAergic. And it turns out that NTS is a good marker even broader than that. Because in this case, we just went to the Allen Institute, the in situ database, and we found another cluster of NTS neurons. In this case, it's again in the periaqueductal gray, the PAG, right? And so when we activated these neurons, so this was done by Peng, Oh, okay. So when we did that, we also saw a huge increase in non-REM sleep, and inactivation causes decrease in non-REM. So these VL-PAG NTS neurons, so these are also glutamatergic. They're also non-REM promoting. Now, we also did optical recording in these PAG neurons, but just for a change, I want to show you a different kind of technique. In this case, we did calcium imaging. Because the PAG, in fact, most of the regions that we look at are buried deep in the brain, so we can't really just do the regular imaging. We had to insert this green lens, which is a microendoscope, so that we can bring the image from deep down here to up here. And we use this miniscope developed by Mark Schnitzer's lab, so that the mouse can actually wear it on the head and move around. And so here is, you can see some cells. And these are the calcium traces. The bottom line is that these VL-PAG NTS neurons are also non-REM active. And so Peng also used the construct developed by Chen Yan to knock down NTS, and that also really reduced the non-REM promoting effect, and knocking down VGLUT2 for glutamatergic transmission also largely eliminated the effect. So again, in this case, both the traditional transmitter and the neuropeptide are important. Okay, so just a quick summary here, right? So we have in our screening for sleep neurons, we found these NTS neurons that label a wide range of populations of neurons. In some cases, are GABAergic, other cases, glutamatergic. They all promote non-REM sleep. And in all these cases, both the peptide and the traditional transmitters are important. So I also want to just very quickly highlight two things, right? So one of them is that NTS seems to be a really general marker. So I showed you CEA, thalamus, and VL-PAG. In fact, there's one more population, which I haven't shown you the data, which I'll show you later, but I'm going to prematurely summarize that, in fact, these are also glutamatergic NTS neurons that also promote non-REM sleep. So NTS is also a very interesting neuropeptide. It was actually discovered in the 1970s, initially as a hypotensive peptide, meaning that if you inject the peptide into the body, it lowers the blood pressure, right? And now we know it also reduces body temperature and slows down the heart. And in fact, all of these things happen during non-REM sleep, right? So it's actually not just sort of a coincidence, just a dumb marker. It actually is really doing something, right, related to autonomic regulation and sleep. The other thing is that it really seems like the ventral region of the plaque is a upstream excitatory sleep center, because we have found multiple populations of excitatory neurons that promote sleep, and they all project to these downstream GABAergic sleep promoting centers. Okay, so I'm gonna try to quickly tell you one more story, right? Again, using this second strategy, right? So I told you about the POA and the CEA, right? And it turns out that this third candidate region that came out of this anatomical screening also is interesting. So this region was characterized by Dantian Liu, another really, really talented postdoc in the lab. So this region is called SNR, the substantia nigra pars reticulata, which is part of the basal ganglia. Now, many of you probably know that the basal ganglia is really important for controlling movement, right? So to understand how these sort of so-called motor control neurons also seem to be related to sleep, we decided to take a closer look at the motor behavior of the mice. So in addition to EEG and EMG, we also added some video recordings in the home cage. And our collaborators in Beijing helped us to use deep learning to train this something called a UNet for image segmentation, right? It seems like, you know, deep learning is all the rage. The whole planet is doing deep learning, so we didn't want to be left out, and that's why we did it, right? So anyway, so you can see that this is doing a pretty good job, right? So this is the original image, and this is where the network thinks the mouse is, right? So basically, once we have this, we can, you know, have a relatively streamlined analysis of behavior. So what we did was to extract two parameters. One of them is a translation. We basically look at the center of mass of the mouse in two different time frames and see how much it moved. The other is total movement, which is the total number of pixels that are different between these two time frames. And when we plotted these two parameters, we basically saw three clusters. The dark green cluster corresponds to local motion, right? The mouse is running around, it's really easy to tell. The second cluster corresponds to all the non-local motor movement, right? So they're not, you know, translating, but they're kind of like grooming or eating or, you know, posture adjustment. The third one is basically immobility. But once we look at the EEG and EMG of this third cluster, there are actually two behavior states, right? So one of them is quiet wakefulness, and the other is real sleep. So here I'm just showing you the EEG delta power and the EMG of these four behavioral states. So EEG delta power, which is basically the low frequency stuff, right? So the higher the power, the less aroused the brain is. Now, as you can see that, you know, one thing I want to point out is that these three greenish states, right, they're all awake states, right? So we tend to think of their difference in terms of their different movements, and you can see a clear difference in the EMG. But there's also a clear difference in their EEG delta power, right? There's also corresponding difference in the brain state. Now, quiet wakefulness and sleep, they're both immobility, right? So we tend to think of the difference mostly in terms of brain states, but in fact, there's also a very clear difference in their EMG power. Now, the point is that, right, EEG and EMG, brain states and motor activity, they're correlated across multiple behavior states, and that's, we think, is quite important. Now, when we looked at the transition between these four states, they're not random at all, right? So when we look at all the trials in which they start out in local motion, they always transition into non-local motor movement. We have never seen the case where the mouse was like running in one moment and bam, they fall asleep the next moment. It just doesn't happen, right? I mean, unless you're narcoleptic, right? So you've seen these YouTube videos like the dog was like running, bam, they fall asleep. That only happens during narcolepsy. It doesn't happen in normal animals. Like we don't do that either, right? So if they start out in non-local motor movement, either they go back to running or they go into quiet wakefulness. They don't fall asleep right away. In other words, if we rank these different states, you know, we put them in a single chain, then most of the transitions are between neighboring states, right? So this is the transition matrix. The diagonal means staying in the same state. And most of the transitions are either immediately above the diagonal, meaning going down the chain one step at a time, or going above the chain one step at a time. The only exception we see is that sometimes they wake up immediately, they go into non-locomotor movement and skip quiet wakefulness. Okay, so this is just behavior, and now let's get back to SNR, right? So it turns out that there are actually two subpopulations of GABAergic neurons. Now, when we injected the Cre-dependent virus into the PV, parvalbumin Cre mice, we mostly labeled the lateral SNR, PV positive neurons. But when we injected the virus into the GAT2 Cre mice, we mostly labeled the medial SNR neurons that are PV negative. So we call these ones the GAT2 neurons. So when we use optical recording to record from the PV cells, so this is just an example, most of them are actually more active during these movement states, right? And then during sleep, they really have the lowest firing rate. So these neurons are actually not sleep active. But when we record it from the GAT2 neurons, they tend to be most active during sleep and less active during these high motor states. And so this is just, I'm just superimposing the population average of these two GABAergic populations, right? So the medial ones are sleep active, the lateral ones are not. Now, when we optogenetically activated the GAT2 neurons, we saw an immediate decrease in locomotion or non-locomotor movement and clear increase in sleep. But when we activated the PV neurons, we saw no effect on sleep. The main effect we saw was to decrease in movement and increase in quiet wakefulness. And when we inactivated the GAT2 neurons, we saw huge increase in movement and decrease in sleep, and PV neuron inactivation has much weaker effect. So we think what's going on is that the PV neurons, right, they're movement activated, right, but their activation mainly terminate movement. So we think what's going on is that this is mostly based on the basal ganglia literature. We think that the PV neurons are important for action selection, right? Because every particular movement that we make, we also need to suppress these other undesired movements, right? So the PV neurons, and that's why when you make each movement, there are PV neurons that are activated to suppress these other ones. But if you activate the entire PV population, you basically suppress all movement, right? But the GAT2 neurons seem to be, they just generally suppress all movement and they're active during the low movement and sleep state. So even though GAT2 neuron activation has a very strong effect on decreasing movement and increased sleep, they don't do this by causing direct movement to sleep transition, right? You decrease one, increase the other, you transition, right? But that's not what's going on. So in fact, when we look at the change in the transitions, right, we never see any case where, so these white ones are the transitions that we don't normally see, right, because they're skipping the stage. We also don't see that with optogenetic activation. Instead, we see the increased transitions in these right above the diagonal transitions, right, meaning going down the chain one step at a time, and and a reduction of these other transitions. So we think that this is important, right, because when we look at this first transition, going from locomotion to non-locomotor movement, we say that's motor control, right? And then you look at the second transition, we say it's motor control. But the third transition, from quiet wakefulness to sleep, we say it's brain state control, right? So we give them different names. But these neurons, they really don't care what we call these, right? All they do is that you activate these neurons, they basically push the animal down the chain, one step at a time, right? So we think that this is sort of an explanation of how these seemingly motor neurons can actually also promote sleep. Now, when we inactivated these GAT2 neurons, we also don't cause any artificial transition. We basically bias the natural transitions in the opposite direction. Okay, so we also looked at the downstream targets of these neurons. The PV neurons project to all these usual suspects, motor control regions. The GAT2 neurons project to these motor regions, but in addition, they also project to the dorsal raphe, right? You see that in this region, the GAT2 neurons project there, but the PV neurons do not. So the dorsal raphe contains serotonergic neurons and also dopaminergic neurons controlling brain state, and also the locus coeruleus with noradrenergic neurons for brain state control. And when we label the subset of the GAT2 neurons that project to the motor part of the thalamus, we also saw their axon collaterals in the dorsal raphe and the locus julius. And when we activated the subset of neurons projecting to the dorsal raphe, we also saw their collaterals in these motor regions. So it really seems like it's the same population of GAT2 neurons that send axon collaterals to the motor and brain state control regions. Okay, so we have a GABAergic population. Now we're going to trace back and look for the excitatory input. So for the SNR neurons, the major excitatory input came from the subthalamic nucleus, which I prematurely summarized, right? So these glutamatergic neurons also express neurotensin. And when we activated these neurotensin neurons, we also saw increase in sleep and decrease in wakefulness. Okay. So just to sum up, right? At the beginning, I said that falling asleep is associated with changes in the brain state and also reduction of somatic and autonomic motor output. So it kind of makes sense for the sleep control mechanism to inhibit the regions promoting arousal and these motor activity. So I told you that for the somatic motor movement, right, we have found the STN, SNR neurons that promote sleep, right? So these are basal ganglia, important part of the somatic motor control system. Now I also told you, right, in the PAG, right, in the hypothalamus, amygdala, and I didn't show you the data, but also in the nucleus of the solitary tract, we have found these neurons promoting sleep. So all of these are important nodes of the central autonomic network. So instead of being separate, as implied by this circuit diagram, it seems like the sleep control mechanism deeply infiltrates these two types of motor control systems, right? So I move these yellow sleep neurons into these motor boxes. On the other hand, there seemed to be a separation between the general state neurons and the specific action neurons, right? What I mean is that the PV neurons in the SNR, they're specific action neurons, but the GAT2 neurons are the general state neurons. Now, the general state neurons seem to send axon collaterals to the arousal system and the motor control system so that they can coordinate movement and brain state. So we think that this kind of view of how sleep is controlled also has implications for this other question, which is why do we sleep? Now, we tend to think that sleep is mostly about changing the brain state, right? Because we lose consciousness of the environment, right? So for humans, we're so, like it's mostly about conscious experience, right? So the question is, why is the sleep control mechanism so tightly linked to movement? Why do we need to shut down movement for a period of time each day? And so to me, this suggests that one fundamental function of sleep is probably for cellular repair, right? Why do you need to rest, right? Probably for some kind of restoration and repair. But what I don't mean is it's only for muscle repair, right? And we know that for athletes, during sleep, they really repair their muscles and it's really important for their performance. But we think that it's broader than that, right? For people in this room, many of us are not real athletes. We're kind of like mental athletes, right? But I think that probably, right, I tend to think that the arousal system is kind of like the central muscles. They need to be repaired as well. So that's something that we're really looking into right now. So these are the people in my lab who actually did the work. The first part is Zhe's work, and she has left my lab, running her own lab in Shanghai. Chen Yan and Dan Qian and Peng did the second part, the anatomical screening. And these three people, I didn't show their data, but really they're the early pioneers in my lab. They joined me about 8 or 9 years ago before I knew how to study sleep. They're really the ones who figured out how to do it. We also have many collaborators who helped us a lot. So thank you.
We have time for some questions.
What do you think about dreaming neurons? Are any of those overlap with dreaming?
Yeah, it's a very good question. So I didn't talk about REM sleep so much, right? I mean, most of the neurons I talked about today are mostly promoting non-REM sleep. So the REM control circuit, the key circuits are probably all in the brainstem, more posterior, in the pons and the medulla. So I didn't talk about the data, but about 5 years ago, we published a paper showing that there is a population of neurons in the ventral region of the medulla. They very, very powerfully promote REM sleep. Pretty much if you optogenetically activate these neurons, you turn on the laser, if the animal is already in non-REM sleep, in 95% of the cases, you can push them into REM sleep within like 30 seconds of activation, right? So those, and this is actually based on earlier lesion studies. If you sort of make a cut in different parts, people have shown that the REM control, the key circuit, is mostly more posterior. So that's consistent with what we found as well. And it seems like the REM circuit is probably simpler. It's not as distributed as the non-REM control circuit.
What do you mean by repair? I think it repairs. When I love sleeping, it repairs my
Good for you.
too. But what do you mean by repair?
So I'm sort of using that term deliberately in the vague sense because I don't know. So if you ask me this question, what's the function of sleep? In fact, everything I talked about is how we sleep, not why we sleep. In my mind, these are really the two big questions, right? So if you ask me like a year ago, say, why do we sleep? I get asked this a lot. Now my answer is that I don't care because all I do is to figure out how we sleep, right? But it's sort of a story of my life. Very often I would say I don't care about something, and then bang, one day, that's the only thing I care about, right? I tend to have these big transitions. So I only started caring about this big time in the past 6 months. So it's way too early to tell. We have a few sort of general ideas. I think that protein synthesis is going to be a big part of it. So one of the things that we already know from human studies is that growth hormones, they're actually, growth hormone is mostly released during sleep, right? So we spend about a third of the time sleep, right? If you sleep 8 hours per day, you're a lucky person, right? So it's a third of the time, but in fact, probably 70% of growth hormone release happens during sleep. Right? And so growth hormone, we know peripherally it stimulates muscle growth and bone growth. It all has to do with the sort of motor activity. But then I think that there's also a central effect as well, because for people who have growth hormone deficiency, obviously if you have that during development, development is affected big time. But there are these cases where people develop that as adult, right? If you have like a tumor in the pituitary gland or something. They not only have, weakness of muscle and stuff like that, they also have, depression and sort of cognitive impairment. So I think that there's that angle in the central nervous system as well. But there's very little in the literature. So that's one thing that we're looking at now. And there are also these, general things like, metabolic stress, right? So I think that that's also a big part of repair.
Thank you.
Luke?
Following the first two questions, I wonder your comments about the function of the REM sleep in terms, because if you observe the EEG signals between rodents during a REM sleep, the state oscillation almost looks like the same as when animal is in awake state in the confusion behavior.
Yeah. No, I mean, it's a very good question. In fact, how important is REM sleep is very controversial, right? I don't know because I only started thinking about function recently, right? So, when I ask people, some people would say that REM sleep is probably not important. And then they say that one reason is that for people with depression, if you take antidepressant, the big class of that drug is before ketamine, right, is a monoamine reuptake inhibitor. So you basically increase the monoamines, right? So it turns out that monoamines system is completely shut down during REM sleep, right? So they're awake up here, non-REM down here, REM is completely down, right? And if you want to have REM sleep, you really have to shut down the monoaminergic system. So if you boost the monoaminergic system, you really reduce REM big time. So people's argument is that, these people, when they start taking antidepressant, they seem normal. They seem to be doing better than before they take the drug. And yet REM sleep is greatly reduced, so that must mean that REM sleep is not important, right? Although when I talk to some clinicians, they say initially it's down, gradually it comes back. So then there's a question of, long-term REM deprivation, does that have some effect, right? It clearly seems like short-term probably not as bad as non-REM sleep. On the other hand, I've talked to a psychiatrist, and when I asked him, I said, is it true that REM sleep is not important? He said, who told you that? If you lose REM sleep, you go psychotic, right? So then I think that the confusion is, part of that is that, sometimes people reach a conclusion, but with different kinds of quality of data, right? When people say the patient completely lose REM sleep, how rigorous is that measurement, right? And unfortunately, I'm not from the human side, so I'm not qualified to evaluate that. But I do know that there is a recent study from a friend of mine, Yuval Nir, in Israel. So he actually tracked down this patient, a famous patient, almost like an HM in the sleep field, right? So he had an injury, like there was a shrapnel or something during a war that lodged in his brain. So, if you measure EEG, he has no REM sleep, very little, right? And that happened like 20 years ago. He's a lawyer, he has a successful practice, right? So you can use that to say maybe he doesn't have, maybe REM is not important. On the other hand, you can say, well, how do you know he doesn't have dream, right? I mean, all you know is that EEG, it doesn't look like REM. So unfortunately, you get into these sort of very complicated, nuanced arguments.
So one last one maybe?
Yeah, you mentioned like the growth hormones and stuff as far as like why we sleep, but like couldn't some of that be circadian? Like I felt the effects of circadian because of spring forward this morning and slept through alarms and stuff.
Yeah.
So I was wondering, like my question is like,
How do you know?
is, like,
Yeah.
sleep causing like setting your circadian cycle, or is this?
Yeah, so it's a very good question, right? So which is why actually sleep and circadian, it's very closely related, right? When I first tell people I study sleep, they say, oh, you study circadian rhythm. No, they're separate, right? Circadian rhythm, it regulates everything, not just sleep, but also feeding, body temperature, everything, right? Except sleep is something that's the most obvious, right? Sleep is separate. So when I say the growth hormone is dependent on sleep, there are actually people showing that, the way you separate those two is, you look at normal sleep and then you deprive sleep, right? And so what people have found is that, by the time you're supposed to go to bed, but if they sleep deprived during that day, actually your growth hormone release is reduced until you allow the subject to go to sleep, right? So there are ways to separate them, but you can't just sort of say, oh, normally it happens at night, therefore it must be sleep, not necessarily.
Okay, well, so let's thank Yang once again.